Most people have their first and last names in here. You may want to, if you don't and you want attendance points, you may want to readjust and kind of leave the room and then change your name and then join back into the room, and that'll change your to your first and last name. That's how we assign attendance points. It's hard for us to assign attendance points with just first names. All right. Well, then we'll just keep it as that. Okay. Well, I think from what I've been gathering through the week, it seems like everybody is kind of at different points within this class, different times, being, chapter 4, chapter 5, chapter 6, chapter 7. So we can talk about a large plethora of things we want to talk about tonight. We could talk about endocrinology. We could talk about cellular biology. We could talk about um, some kind of transcription, translation, what happens when that goes wrong. We can kind of take it anywhere. Does anybody have a topic that we want to discuss? Anything in the laboratory? Anything in endocrinology based? Because we've been doing a lot of that recently. Negative feedback, positive feedback, anything along those lines. Okay. So it sounds like we may be a little bit quiet tonight. That's all right. This will likely be a short spotlight session then. Um, what I do want to make sure we cover is that we understand negative feedback loops and positive feedback loops. Um, oh yeah, thank you, Cassandra. Yeah, we can talk about that. So I'm glad you brought that up. So what we're asking is that we're really trying to figure out what's the best way to retain this information, because as we know, it's a lot of information, right? We're moving fairly rapidly to cover a broad base of information, and typically you get that. Um, that's okay. I mean, C's are okay. We can probably definitely bump that up. We just got to tweak a few things, right? And Jennifer certainly agrees, and I do too. So it is a lot. This is an introductory level class where we literally give you a round the world view of every topic, really, in biology, physiology, biochemistry, pathophysiology, cell biology, everything, even histology, study of tissues. Um, so it can be sometimes tough to keep up with. And what I've told people in the past, and this is kind of what, and I've even heard students say this too. Um, I will say mine first. Yeah, Shelby says there's a lot of lab and lecture information, and yes, there is. We really have to look at this material every day. Okay, we have to look at this material probably every day until we get comfortable enough where we maybe can take a day or two off. That doesn't mean that we get burned out, okay? Because certainly by the time November runs around, I'm even me as an instructor role, I start to feel burned out, okay? And you just got to press through it. So what do we do? Well, we need to spend time with the material, okay? Spend time with the material. My TV's off, my cell phone's charging. It's me and the computer, me and you guys, right? This is what I would encourage your surroundings to be like too. It's also quiet. Okay. Now, a lot of us have kids. I got a couple cats running around that like to play at night. So I have actually earmuffs or gun muffs that I put on to silence everything so I can concentrate. I would try to encourage you to find something along those lines. I'd also, and it's tough, it's not going to come easy. You can't just sit back in your bed and read the book and think it's going to stick. What you really need to do is sit at a desk or sit at the kitchen table or whatever you do and get a piece of paper out and get a pencil out or a pen and start rewriting your notes in your own words. Okay, it's very important that you rewrite the notes or the PowerPoints in your own words, because what that is really doing is that it's telling your brain that this is what negative feedback inhibition is, or this is how transcription works. And you literally tell a story on a sheet of paper of what transcription translation is. And then you can fact check this based on the PowerPoints or the textbook. All right. It's not for the faint of heart. It takes effort. It takes time. You know, your fingers may be a bit cramped. It may take a long time, but you really have to try to. That's what I would recommend. Okay. Now there is some pros to this. Is that all of a sudden you have a study guide. Okay. You've rewritten your study guide. All right. And then all of a sudden you can start circling things, highlighting things on the study guide to see where your soft spots are. Right. You can, and if you start early enough or you follow along with the lecture, then maybe by the end of the week on a Saturday or Sunday, you may want to go back and review what you've written down that week and build upon it. What are the take-home notes here? What did the professor or the instructor emphasize? You know, what was the case study about this for? And that's kind of where you start really honing in on high yield material. Okay. Um, some questions on the first exam will be just straight rudimentary definitions, right? Like an arrow to this or an arrow to that. Those are easy points. Some of them are a little bit different. I know with the lab exam over the tissues, if you don't answer question 1 right, you may not be able to answer question 2 or question 3 correctly either because you didn't know question 1. So some of it builds on each other, and that could be 6 points. You screw up 2 of those, you know, maybe you get credit, half credit, it's 10 points, 9 points. All right, that's letter grade. So it's kind of important that little baby stuff, it's important that we get the easy things right. All right, so the easy things, in my opinion, are memorization. Okay, anatomical position. What's dorsal, what's distal? Don't get those confused. Anterior versus posterior. That's the easy stuff. That's the low-hanging fruit that we should be able to hit out of the park and get that 2 points. If you don't, then, you know, that's maybe why your B turned into a C. Okay. And that doesn't mean we have to master every hard concept. All right. If you find yourself studying and you're just like, screw this, I don't understand this, and it's sucking up all your time, then the hell with it. All right. Let's roll the dice. Let's say there's a chance that there is maybe 1 or 2 questions on there and we don't know it. You got a 25% chance of making it. Then I would just move on. You know, don't get bogged down on one concept. If you do, then you probably need to come to Spotlight session and share it with me. Okay. Because then I could probably help straighten this out, iron out the wrinkles real quick. All right. And then we can have a conversation about it. We had a really good conversation last night that went to 9:58, had a full hour. And I think students were just blown away by how good they felt on a few things. So we're kind of off to a slow start tonight, but hey, you know, whatever. So yeah, we can, Caitlin, definitely. Let me finish up this one concept. So that's my thing. Spending time with the material, writing things down, and having your study guide. Yeah, sure, Brianna, definitely. We'll put that on the list. Thank you. Other students have had really good luck with Quizlet. All right. Quizlet. Some of us may know, some of us may not know. I studied Quizlet when I was a student. I just typed it in there. So I'm not sure what it is or where they got it from, but they were loving Quizlet. Quizlet to me is more of a memorization tool. I don't think it'll carry you very far, particularly in the higher levels. But yeah, see, Cassandra says she uses Quizlet and YouTube crash courses. Okay, so I guess in that case, that was your best way of retaining the information. Maybe so, maybe not. I'm not sure, but it works for some people. I would want students to write out their notes and use it as a study guide. Flashcards are cool. Yeah, I mean flashcards are cool. I'm not a big flashcard person. If you've kind of been in these before, I kind of dog flashcards. They're good for anatomical position. They're good for learning the bones. I don't think they're very good for conceptual stuff. But then again, this class doesn't really go into much detail, believe it or not. So maybe you can get away with flashcards and at least helps you narrow down some of the choices, and voila, you got what you needed. So let's see. So yeah, Jennifer says she's had good luck with Quizlet. Jaden has, and Cassandra has. Okay, so that's kind of my spill there. Laboratory, yes, laboratory will complement lecture. Okay, so sometimes you'll get a little bit of dose of both concepts, which could be good. Like diffusion. All right, thanks, guys. As we all know, that happens from time to time. Justin, you're always quick on the draw there. I appreciate that. Let me get my video going too. So what we want to do is, let's see. Yeah, you can. I mean, 74 is not a bad start. We can definitely build on that. You want to make sure you get the easy points right, the assignments, the homeworks, the quizzes. Like we don't want to miss those. That's cake. That's cake points. Okay. You don't want to shoot yourself in the foot screwing those up. The skull for Shelby, that is just flashcards. That's quintessential flashcards. That is Quizlet in my opinion, because you're just it's just rapid memorization, recall, right? This is the nasal bone, this is the frontal bone, this is the mandible, this is the mandibular bone, that kind of stuff, xiphoid bone. And yeah, there you go. See, Kennedy found a good. See, I mean, that's a perfect grade. I'm sure you were tickled when you saw that. Used expo markers to write them out. See, keyword there. Write on them and study it. Yeah, yeah, you wrote them out. I want to emphasize that. So I think that answers your all's questions right there. And if it doesn't, chat me something we can elaborate. So this is the point where I'm going to kind of stop getting hunched over. Eventually, I'm going to have my office set up in this house, and I'll be able to have a better setup. But until now, I'm on the couch. All right. Let me just let's get going here. Let's get back to these topics we wanted to get on. So Caitlin brought up DNA and RNA. And DNA and RNA and translation. So let's take it from the top, right? When we have DNA replication, we have chromosome that kind of gets unwound, right? Because chromosomes is pretty much condensed DNA strands. We really can't do much DNA replication or transcription in this kind of form. So this is maybe when mitosis comes around, right? When we start to kind of unravel the chromosome, we kind of get these spaghetti-like strings of DNA, which all of a sudden makes these areas accessible, right? Now, we love to use the word expression or expressed. Something is expressed. What do we think that is? This took me forever when I was a student to figure out when something is expressed, what does that really mean? Throw me an answer in the chat. Yeah, it's shown. Let's dig in. Let's do a little bit more detailed, Sophia, if you don't mind. Yeah, it's there, it's available, but like, what is it? Like, what are we doing there? Thought or feeling. I'm not sure if I'm following you, Kennedy. I just want you all to give me what do you think is happening when something is expressed, when a gene is expressed? Maybe a little bit tougher than what we think. All right. Okay. Yep. Seth. Yep. Phenotype. So Seth is getting pretty warm there. And Sophia says it's in progress. So I'm going to go ahead and take over here. When we are in progress or we are changing the phenotype, what we're really meaning is that if a gene is being expressed, that means that we have RNA polymerase on that gene creating mRNA, which then that mRNA becomes mature mRNA, goes out into the cytoplasm, and then all of a sudden tRNA comes in and then starts reading off the mRNA to make protein. So essentially, when something is expressed, that means that gene is being transcribed and then translated to make an effective protein, an active enzyme, an active hormone. If you remember, enzymes, hormones, they're all the same thing. They're a protein. They're just different classes of a protein. A hormone travels very far that can bind to a receptor and carry out a function within the cell. A enzyme is a protein that carries out a reaction, right? We'll get to that next. And let's see, who brought up, Brianna brought up ATP in the Krebs cycle. Well, that's what enzymes do. Enzymes are the Krebs cycle. So we'll talk about that next. So let's make sure we know when we're talking about, if you hear somebody saying that this is expressed, it's showing its phenotype. That protein is actually the phenotype. What is a phenotype? A phenotype basically is how something appears. My phenotype is I have green eyes with brown hair, with now a receding hairline, right? That's something new. It's not as bad as my cousins, but it's there. So that is my phenotype. My genotype, because let's just finish out this concept here. Genotype with a G is basically the genetic makeup that is my brown hair, green eyes, and receding hairline. Okay, that's what's considered the genotype. So if I'm growing brown hair, then that means the gene that codes for the protein that makes up my brown hair is being expressed. That means we're going to make mRNA on that brown gene, and then we're going to make the protein from that brown gene. And that's kind of how we get brown hair. So that's what we mean when something's being expressed. But back up to Caitlin's point, we have DNA, we've loosened it up because let's say we're in mitosis and we got to cut these big spaghetti DNAs. Yeah. That's correct. Marilda, I hope is how you pronounce your name. Excuse me. Yes, it's showing its purpose. That's why it's there. We don't show the purpose of these genes all the time, right? We don't express everything. Um, uh, Puberty genes are only expressed during, ages 12 to 14 or whatever it is, right? There's different stages in life where we may express a gene. Babies, growth, when we start to walk, when we start to talk, developmental things. These are key areas when we express certain genes at a specific time that help our development. So those are prime examples. So now we've loosened up the DNA. We have the these spaghetti DNA strands, which pretty much means that RNA polymerase, which is an enzyme because it has that ACE on the end, can bind to the DNA and start cranking out mRNA, right? It's matching up, it's replacing the thymine with the uracils, and all of a sudden we have this long nucleotide strand with uracil, which means that it's mRNA, right? So mRNA, as we may or may not remember, has introns and exons that kind of make up the mRNA. And then what we really need to do is that we need to get it out of the nucleus, and then we need to send it out to the cytoplasm. So in order for us to do that, we have the nucleolus, right? We have the nucleus, and then we have the nucleolus, or I should probably say the nuclear envelope. Let's change that over to the nuclear envelope, which is kind of like an inner membrane that has these gates. And in order to get the mRNA out of the nucleus and into the cytoplasm, it has to be what we call mature mRNA, not pre-mRNA, mRNA or mature mRNA. So on the 5' end, which is the left-hand side of the mRNA, it's going to have a guanosine cap, otherwise known as a G cap. Think of it as like a flag or a signal that says, hey, I'm old enough to come out to the cytoplasm. And on the 3' end, or the right-hand side, it's going to have a poly A tail, or a bunch of adenosine nucleotides attached on the end together, which is called a poly A tail. And then once we make it out to the cytoplasm, then we're ready for translation, right? We're ready for translation. And so what happens is that this mRNA, this mature mRNA, it's also spliced out, cut out, or spliced out its introns. We have no introns in mature mRNA. They don't do anything. So we have a G cap, a bunch of exons, and a poly A tail. That makes up mature mRNA. And what happens is that once we've made it out into the cytoplasm, then we start this translation, right? So we have tRNAs come in, which is another type of RNA. It's called transfer RNA. And they will read 3 nucleotides together, right? And those are called codons. So you can think of it as like 3 fingers here, and they'll read the first nucleotide, the second nucleotide, and the third nucleotide. They read them in triplets, yeah. Not triplicates, triplets. We call those codons, right? So it's kind of more specific. And the thing about these tRNAs is that they have something they call charged tRNAs. Does anybody know what a charged tRNA is? Question marks? I mean, is anybody even listening? If we don't know it, just say we don't know it. I could have protons, yeah. So, yeah, so Monisha said amino acids, and she's right. Now, to Cassandra's point, Cassandra probably zoomed in a little too much. There's all kinds of protons on these amino acids. All kinds of protons. We have protons, gobs of protons. Those are like down to like the molecular, the very small atom size. We want to zoom out a little bit and say that those are amino acids. Amino acids have protons on them. So, so we have these amino acids, and when we match up our codons to their complement on the mRNA, it will deposit an amino acid in what's called the P site or processing site. The ribosomes, I always forget the ribosomes. The ribosomes are really what attach onto the mRNA. Okay, that's the purpose of ribosomes. They will go and they will find this mRNA that's kind of floating out in this cytoplasm. And what happens is that the ribosome has 3 sites. It's called EPA: exit, processing, accepting. So the tRNAs will come into the accepting site and see if their codons match up with the mRNA codons. If they do, it moves to the P site, and then it will deposit its amino acid right there. Typically, the first amino acid in any protein is going to be methionine. That's what starts it. So all of a sudden, the methionine charged tRNA is going to look for AUG, adenosine, uracil, guanosine, AUG. And as soon as it finds that, it says, "Oh, this is where we start making the protein at this particular point in the mRNA. This is when we start to express, right?" So then it deposits the methionine right there at the P site, and then it gets kicked out on the exit site. All right, so it comes in right, and then kind of moves to the left, and then it goes out. I always remember it as EPA, Environmental Protection Agency. Now kind of help you remember what the purpose is. So then, we have 20 of these tRNAs because we have 20 amino acids. And then they will come in and they will see what fits. And then if something fits, then they'll make a deposit on their on the P site, and then you'll build out this protein. Eventually, you'll reach what's called a stop codon. And the stop codon is like UGA, University of Georgia, UAA, and then there's another one. I can never remember the third one. Anyways, when that happens, that means that we have reached the point of the end of expression with that mRNA, and then eventually we will cut this protein off from the ribosome, and then some magic happens to where we start to see this protein fold to what we call its native form or its conformational form. As you may recall, we probably have polar and non-polar amino acids. Polar means water-loving. Polar means hydrophilic. Same thing. Polar, hydrophilic, it's the same definition. We also have non-polar amino acids, water-hating, or hydrophobic. Amino acids that are polar will be found on the outside because we're made up of a bag of water. So it will like to interact with water and other polar elements. And all the non-polar amino acids will find their way to the inside of that protein. Now, whether that's a hormone or that's a enzyme, depends on what we're really expressing on our mRNA, which really means what we're expressing on our gene, which is DNA. So that is kind of the concept where we go from DNA all the way down to protein, and we're expressing something. So I'm trying to remember who asked that question. Might have been Caitlin. Tell me what you think about that. If we need to clean something up or tweak something, please let me know. And then we'll kind of move on to the next question, I think, was Brianna's question about metabolism, essentially. Okay. Okay, cool. Thanks, guys. So Brianna's question really talks about metabolism, and this is biochemistry to the heart. Biochemistry is a complicated subject. It is a very interesting subject. It's kind of like one of my wheelhouses. So we've talked about expressing proteins, and sometimes the genes that are expressed will code for enzymes. If you remember what enzymes are, they actually carry out a reaction. All right, they carry out a reaction and they take something and turn it into something else. So when we have these concepts about enzymes and stuff like that, what we're really trying to do is we eat food to extract energy from it, right? And the reason that, or it just so happens that the energy that we store, the molecule that we store energy in is ATP. It's very stable. We can easily break it down and release that energy to live our lives. So the reason why nature has chosen to place our energy into ATP is for that reason. It's very stable, and it can hold a lot of energy. So 44 ATP, or I can't think of that. It's not coulombs. It's something else. Calories, like 44 calories, or not, maybe it's 19 calories, something like that per ATP, which is kind of neat. So when we eat dinner or we eat donuts, I kind of gave this example a couple nights ago. We have what we call polysaccharides, right? Complex carbohydrates that we really can't use yet because we have to break them down. Catabolism, breaking things down, versus anabolism, which is bringing things up. But in this case, we're in the business of breaking things down. And when we break things down, what we really want to do is try to get it down to monosaccharides, otherwise known as glucose. Glucose is where we can really start to do stuff with. Glucose, once we get it down to glucose, we can start glycolysis. Glycolysis, and before we get there, enzymes really break down these carbohydrates. Somebody brought up what is amylase? Well, amyloid is basically sugar or starch. Amylase, ASE, that is an enzyme that breaks down starch, right? That's actually secreted in our salivary glands in our mouth. So if we're eating pizza, we're already breaking the pizza down in our mouth before it enters our stomach, which is kind of crazy. Because then we start to absorb the glucose very quickly. So when we get it down to glucose, we can enter glycolysis. And I don't want to go into too much detail because I don't want to lose you guys, because you could spend a semester learning about metabolism. But essentially, we go through these different steps using these enzymes. It's 10 steps. And what pops out at the end of glycolysis is a molecule called pyruvate. And pyruvate is a 3-carbon molecule. So just to give you an idea, the whole world's made up of carbon. Carbon is everywhere. So glucose is made up of 6 carbons. So it's a 6-carbon structure, think of it as like a little square, or something, polygon or whatever it's called, hexagon, 6, right? Let's think about that. So think of it as like, and so then we break it down to 3 carbons, pyruvate. Now think about pyruvate, and let's not get caught up too much on some of the vocabulary, but I just want to take you through this. Pyruvate can go anywhere. Pyruvate can make fat. Pyruvate can make protein. Pyruvate can get broken down further into ATP. It's basically whatever the body needs. We can shove pyruvate in some different process, either direction. And that's kind of how we can, use what the body needs. So if we're still wanting to break down pyruvate into ATP, we send it to the citric acid cycle. And what we're really doing in the citric acid cycle is we're taking a number of steps of enzymes, and we are extracting protons from it. Kind of what Cassandra was saying earlier. This is when we're kind of getting into the nitty-gritty. We are taking out protons in each of these molecules because we're going to use these protons. and we're storing them on a molecule called NAD to make NADH. All right, if you remember, hydrogens are also called protons, because if you look on the periodic table, hydrogen essentially is made up of one proton because it has 1 as the atomic number. The atomic number is essentially the definition of protons. Helium has an atomic number of 2, so that means it has 2 protons. So when you see a bunch of hydrogens tacked on, you may hear somebody say it has a bunch of protons tacked on. So you'll hear me use protons. It's the same thing as a hydrogen or an H. Okay, make sure you follow that. So then all of a sudden we're taking all these protons or all these H's and we're sticking them on a molecule called NAD to make NADH. All right. And all of a sudden we got all these NADHs that we've mined or we've extracted from pyruvate. And what happens is that all these hydrogens go into the mitochondria, the intermembrane of the mitochondria. And we will take these hydrogens or these protons and we will put them into the proton ATPase pump. And I'll skip the details, but as these protons get pulled through this pump, similar to like the ATPase pump, sodium potassium ATPase pump, this is a proton pump. And as these protons get pumped through into the mitochondrial matrix, which is the innermost part of the mitochondria, it takes enough energy to place a phosphate back onto ADP. So we have just this storage of ADP, adenosine diphosphate, used up ATP. Remember, if we want to break off some energy, we take ATP and we all of a sudden we break a phosphate off and we have ADP. Well, we just cycle those through. So then all of a sudden we take that ADP, we put it right next to the proton pump, and we use that energy pumping that proton through to place a phosphate onto ADP to make ATP. And then we send that ATP wherever it needs to go. And that's essentially how we make ATP. There's other ways we can make ATP, but the large way is breaking down glucose. So, Brianna, what do we think about that? Or does anybody else want to have a comment or a question? Sounds like we have a bunch of silence. Okay. Okay. All right. Well, thank you, Kayla and Brianna. I appreciate that. I've always told students in the past, if you really want to listen to this again, last year I made up a YouTube account, and we can always post it on YouTube once it uploads. I have a few of them up from this semester, and if this helps you, you want to listen to it again, I'll throw it up on YouTube, and we can do that. Okay. You want to, Caitlin? Okay. Okay. Kayla says yes too. Okay, we can do it. So I have to. Yeah, I agree, Memphis. That's why we're here. That's why we're here. Okay, guys. All right, cool. We can do that. So it has to upload to this system, and then eventually I can upload it to you. To download it, upload it, blah blah blah. So when you're looking for my stuff, you just type in bio 130. Yeah, type in bio 137 Mattmiller spotlight session. Okay. Somebody actually wanted me to get the links, and I actually got that the other night. So bear with me. I could get you the link. Hold on. I guess this is what people call my channel. All right, that's my stuff. All right, Kennedy found it. Thank you. It's not much there. I mean, this is kind of basic. So I can get this up to you all after we're done here. Yeah, I agree. I agree. It's better to listen than it is to read about it because you have to really picture it in your head. Okay. Let's see. Are we done? Is there anything else we want to touch on? All right, good. Jaden got it. Kayla's happy. Kennedy's happy. Diane agrees we need to do YouTube. So does Sarah. Lativia says that too. Okay. Gotcha. Are you posting this one? I can, Kennedy. If you want me to post last nights, I can. That's how I learned, Amanda. I recorded all my lectures and then I rewrote my lectures when I was a student. Yeah. Okay. Okay. We can do both. Let me find it. LaQuisha, I'm Monday through Friday, 9 p.m. Yeah, I am too, Amanda. I am too. Sometimes I'm a little bit of both. You got to figure it out. Yeah, no problem. Keep signing up, keep coming with questions, and then, everybody benefits when we have questions. All right, good, good. I'm glad this is worth your all's time. All right, it sounds like we're good on questions tonight. So let this download to the system, and we can go from there. Yeah, sure, Seth. Hold on. I like off the wall questions. Since glucose creates pyruvate and pyruvate can create proteins, can a person create muscle mass with strictly carbohydrate diet? Probably not, because you would have to use up all of your glycogen, which is your glucose stores, and then no, because protein's really first to go. That's the thing. Like if you were to go on a diet, like you're going to use up some protein, and then you're going to use up some of your glycogen, and then you're going to start burning your fat. Like the keto diet, that's just all meats and fats. That's what you're trying to do. What you're really trying to do is burn up all your glycogen, which is your carbohydrate store, and then you really want to like catabolize your lipids and your fats to make energy. It's fine, it works, but it also produces high cholesterol, and that's not good for cardiac. I don't know. Because I moved to burn glucose, I love. Yeah, carbs. I don't think carbs. And see, the thing is about carbs is that you can eat a lot of vegetables and it can stick with you the whole day. But if you go eat a donut or have this carb crash or whatever, like yeah, you'll feel great for an hour or two, and then those carbohydrates just get burnt out, and then you feel this drop, and that's why carbs are never very good. But if you eat chicken or rice or vegetables, you can feel full for a whole day, and that's just because they burn a lot slower. They have a lot more energy per or more calories per gram of substance, I guess is the lack of a better term. And that's kind of what gets interesting in the biochemistry is the kinetics of it. What glucose will yield versus a gram of glucose versus a gram of lipid. The amount of calories you get per macromolecule. So that's kind of neat. But no, Seth, I don't think so. I don't think you could. The only thing you're building on a strictly carbohydrate diet is you're going to be a fat ass. and that's lack of a better term. So you're going to build up a lot of glycogen. Yeah, yeah. So you're probably going to be diabetic at one point because the insulin, if you think about it, we have a lot of carbs and sugars, and then the glucose transporters that we talked about a few weeks ago on the cell membranes, they get desensitized because they're being used up so much. We can't have a bunch of glucose hanging around in the bloodstream. That's how you become diabetic. It damages the kidneys, damages the brain, does different things. We need to have that glucose stored somewhere, whether it be glycogen inside our cells and stuff like that. But if we continue using these glucose transporters, they get desensitized, and then that's how you get type 2 diabetes. Yeah, they do it for energy, I think. They do that. I mean, carbs would be for energy, but what you would probably want to do is you use the carbs to build up the glycogen. Like if my wife goes and runs a half marathon, she may want to go eat a bunch of whole wheat spaghetti, right? Because the whole wheat spaghetti will then get broken down into glucose, which then gets stored into glycogen. And she will use that glycogen to keep up her blood glucose levels. Eventually, maybe halfway through her run, she'll convert over into keto or fats, right? But maybe some protein, it just depends. I mean, your body can't break down too much protein because it's using it in your muscles. So it's going to want to probably divert over into the keto form, which is fats. That may be a better answer. Okay, so it sounds like we're good. We can talk about proteins and all that, what happens when we have diabetic ketoacidosis. Probably can't do it tonight. Or protein, sometimes what happens, with the stomach and proteins. We'll say that for another night. All right, guys, we will get last night's YouTube up and tonight's YouTube up, maybe a half hour. So, but I'm going to work on it, and we'll have it up probably before 11 o'clock, hopefully. Yeah, no problem. All right, let's call it for tonight. We'll see you all tomorrow or another time. If I don't catch you tomorrow night, see you next week. All right, yeah, Brianna, Memphis, Kayla, Caitlin, Kennedy, thank you guys. Always helps when we have participation. I appreciate it. Alicia, Monisha, Jaden, Amanda, Jennifer, Lativia, Shelby. All right, guys, take care.